Depth 109 or 28 meters. At bottom.
At nearly 36,000 feet below the Pacific surface, where pressure exceeds 1,000 atmospheres and sunlight has never reached in all of recorded biological history, cameras descended into what scientists once described as a near-certain dead zone. Unexpectedly, they captured movement. This is the deepest known environment on Earth, the Mariana Trench. For decades, the assumption was that below a certain depth, complex life should collapse under the physics of pressure and energy scarcity. Yet, footage from the Mariana Trench revealed something alive at 36,000 feet, and it shouldn't exist. This is one shocking dive you wouldn't want to miss. Into the deepest place on Earth. 200 miles east of the Mariana Islands, the seafloor suddenly breaks apart and drops into the deepest scar on Earth. The Mariana Trench stretches for roughly 1,580 miles across the Pacific, but its most infamous point lies near the southern end, Challenger Deep. At nearly 10,900 meters deep, or about 36,000 feet, it descends farther below sea level than Mount Everest rises above it. If Everest were dropped into the trench, its peak would still sit more than a mile underwater. Down there, sunlight disappears entirely. The darkness at Challenger Deep is older than humanity itself. No ray of sunlight has touched that environment for hundreds of millions of years. The trench exists because the Pacific Plate is slowly sliding beneath the smaller Mariana Plate in a process called subduction. The movement is painfully slow at just a few centimeters per year, but over geological time, it carved a canyon so deep that it changed ocean science forever. Temperatures at the bottom hover just above freezing, usually between 1 and 4 degrees Celsius, yet cold is not the real danger. Pressure is. At Challenger Deep, the water pushes with more than 1,000 atmospheres of force. That is roughly 16,000 pounds per square inch. A human body exposed directly to that environment would be crushed instantly. Ordinary submarines would implode long before reaching even half that depth. Every square inch of metal, glass, wiring, and electronics must survive the equivalent of several fully loaded Boeing 747s pressing down on a surface the size of a dinner plate. For decades, scientists assumed almost nothing complex could survive there. The deepest ocean zone, known as the Hadal Zone, begins around 6,000 meters below sea level. Below that threshold, conditions become so extreme that marine biologists once imagined the seafloor as a barren wasteland scattered with only a few starving scavengers. The logic seemed reasonable. Photosynthesis becomes impossible below roughly 200 meters because sunlight cannot penetrate farther. Without sunlight, no plants can grow. Without plants, the food chain collapses. The only nutrition reaching the trench would be dead organisms drifting downward from the surface, a slow rain of organic debris known as marine snow. By the time most of it sank 7 miles, scientists assumed almost nothing useful would remain. The first clues about the trench emerged during the voyage of the British survey ship HMS Challenger in 1875. Using weighted sounding lines lowered into the Pacific, the crew recorded a depth of nearly 8,200 meters, astonishing for the time. The location was later named Challenger Deep in honor of that expedition. But 19th century technology could only measure the abyss, not observe it. For the next 85 years, the trench remained more rumor than reality, a black void beneath the Pacific that nobody could actually see. That changed on January 23, 1960. The missions that survived the pressure. On the morning of January 23, 1960, 2 men sealed themselves inside a steel sphere barely wider than a small car and began descending toward a place no human had ever reached. Jacques Piccard and Don Walsh climbed aboard the bathyscaphe Trieste near the Mariana Trench with one objective: reach Challenger Deep, nearly 11 kilometers below the Pacific surface. At the time, the mission sounded reckless. Engineers could calculate the pressure mathematically, but nobody truly knew how metal, seals, wiring, and human nerves would behave under more than 1,000 atmospheres of force. If the vessel failed, implosion would happen faster than the human brain could process it. Trieste itself looked strange even by submarine standards. Built originally in Italy and later acquired by the United States Navy, the craft combined a massive gasoline-filled float with a tiny pressure sphere suspended beneath it. Gasoline was used because it is lighter than water and relatively incompressible at depth. The crew compartment was a steel sphere with walls nearly 5 inches thick and 2 small observation windows made from specially engineered plexiglass. The descent took 4 hours and 47 minutes. As Trieste sank deeper, the Pacific outside transformed into absolute darkness. Then came the sound every deep-sea explorer fears, a loud crack reverberating through the hull after one outer acrylic window layer fractured under pressure. Picard later admitted the noise felt like a gunshot inside the cabin. Despite the scare, Trieste continued downward. When the vessel finally touched the trench floor at roughly 10,916 meters, the crew entered a place no human eyes had ever seen. But the moment was chaotic rather than triumphant. Their landing stirred up massive clouds of fine sediment, turning the water outside into what Walsh later described as a blizzard. Visibility collapsed almost instantly. The pair could barely see beyond the windows. They remained on the bottom for only about 20 minutes before beginning the ascent. Although they reported possible sightings of living organisms on the seafloor, the observations were too brief and obscured to revolutionize biology. The mission proved humans could physically survive the descent. It did not yet reveal what truly lived there. Then the silence began. For 52 years after Trieste, no human returned to Challenger Deep. The problem was not courage, it was engineering. The Hadal Zone, which sits at ocean depths below 6,000 meters, became notorious for destroying technology. Pressure attacks everything. Metal fatigues, electrical insulation compresses. Tiny leaks become catastrophic failures. At nearly 11 kilometers deep, even a microscopic weakness can collapse an entire machine instantly. Deep-sea exploration turned into a slow war between human engineering and the physics of the planet itself. Japan emerged as one of the major players in that war during the 1990s. In 1995, the Japanese remotely operated vehicle Kaiko successfully reached Challenger Deep. Operated by the Japan Agency for Marine Earth Science and Technology, Kaiko became the first robotic explorer to conduct detailed scientific surveys of the trench floor. Kaiko could collect sediment samples, record footage, and remain operational. Between 1995 and 2003, it completed more than 250 dives, many at extreme depths. Scientists finally began obtaining serious data from the Hadal Zone instead of isolated glimpses. Then, in May 2003, Kaiko disappeared. During a typhoon-related recovery operation southeast of Shikoku, the vehicle's tether cable snapped. Most of the robot sank permanently into the Pacific. The loss was devastating. Kaiko had cost tens of millions of dollars and represented one of humanity's few reliable ways to study the deepest ocean. The pattern continued. In 2009, the American hybrid remotely operated vehicle Nereus reached Challenger Deep, carrying high-definition cameras and advanced scientific tools. Developed by the Woods Hole Oceanographic Institution, Nereus represented a major leap in deep-sea robotics because it could operate both autonomously and through fiber-optic control links. But on May 10, 2014, while diving in the Kermadec Trench near New Zealand, contact with the vehicle suddenly vanished at nearly 10,000 meters depth. Investigators concluded that Nereus had imploded under pressure. Pieces of debris later floated to the surface. Another machine lost to the abyss. Ironically, the breakthrough did not come from billion-dollar submarines. It came from relatively simple devices called baited landers. These systems worked on a straightforward principle: attach cameras, lights, sensors, and bait to a weighted frame. Drop it into the trench. Leave it there for hours, then release the weights so the system floats back to the surface. The man behind this transformation was Alan Jameson, the footage that changed marine biology. When the first modern baited landers began returning hours of uninterrupted footage from the Mariana Trench, many marine biologists expected long stretches of emptiness. The Hadal Zone had spent decades being described as a biological desert where life survived only in scattered, energy-starved pockets. Researchers assumed that if a camera reached the bottom of Challenger Deep and waited quietly, very little would happen. Instead, some of the earliest recordings triggered a complete reassessment of deep ocean ecology. Within minutes of the cameras touching the seafloor, movement appeared out of the darkness. The first arrivals were amphipods. At first glance, they resembled ordinary shrimp, but these creatures behaved nothing like the sluggish scavengers scientists expected to find 7 miles underwater. The bait, usually pieces of fish or mackerel attached to the lander, had barely settled before amphipods began swarming it in huge numbers. Some species measured nearly 30 centimeters long, enormous for deep-sea crustaceans. Their bodies flashed pale white beneath artificial lights as they piled over one another in frantic feeding frenzies. Researchers watching the footage realized something was wrong with the old model immediately. In an environment supposedly defined by starvation and low metabolism, these animals moved aggressively and fast. The deeper scientists looked, the stranger the trench became. Expeditions led by Alan Jameson recorded amphipods arriving so quickly that some researchers suspected they were detecting vibrations or chemical signals from remarkable distances. At depths beyond 8,000 meters, the animals did not drift lazily toward food. They rushed it. In some deployments, bait became completely covered within hours. One reason this shocked marine biologists was the temperature. Water near the trench floor typically sits between 1 and 4 degrees Celsius, conditions expected to slow metabolism dramatically. Yet the amphipods behaved more like competitive scavengers from shallow ecosystems than creatures living under 16,000 pounds per square inch of pressure. Scientists began realizing the Hadal Zone was not populated by isolated survivors barely hanging on. It contained functioning ecosystems. Different species repeatedly appeared together in recognizable ecological patterns. Some organisms scavenged aggressively, while others sifted through sediment for organic particles. Tracks and feeding trails marked the seafloor mud. Creatures interacted with one another rather than existing as random, isolated specimens. The trench floor showed evidence of food webs, nutrient cycling, and ecological specialization. That was the true shock. The deepest environment on Earth was not simply occupied, it was organized. Among the most common trench dwellers captured on camera were deep-sea sea cucumbers, members of the holothurian family. Unlike the colorful tropical species familiar to divers, Hadal sea cucumbers often appeared ghostly and translucent, crawling slowly across the sediment like drifting organs pulled by invisible currents. Species such as Elpidia and scotoplanes moved constantly, vacuuming organic material from the mud. Some scientists nicknamed scotoplanes sea pigs because of their swollen bodies and stubby leg-like tube feet. Their presence revealed something important. Enough food existed on the trench floor to support large populations of deposit feeders over long periods of time. Then there were the xenophyophores. At first, researchers mistook them for rocks or strange sediment patterns scattered across the seabed. In reality, xenophyophores are giant single-celled organisms, some reaching more than 10 centimeters across, making them among the largest individual cells ever discovered on Earth. These delicate life forms build intricate structures from sediment particles collected around them. In the trench, they covered sections of the seafloor in surprising density. Their importance went beyond their bizarre appearance. Xenophyophores create microhabitats for smaller organisms, essentially functioning like miniature ecosystems themselves. Even at Hadal depths, life found a way. Then there were microbes. High-definition footage and sediment samples revealed microbial mats coating rocks and mud in parts of the trench. These mats contained dense populations of bacteria and archaea capable of surviving under immense pressure. Some researchers suspected certain microbes might rely partly on chemosynthesis, deriving energy from chemical reactions instead of sunlight. That possibility carried enormous implications because it suggested the trench food web might not depend entirely on organic matter falling from the surface ocean. Deep below the Pacific, biology may have developed alternative ways to sustain itself. The trench also appeared far more dynamic than earlier models predicted. Organic material did not drift downward evenly like gentle snowfall. Large food falls, dead fish, squid, and whale remains occasionally plunge into the Hadal zone, delivering sudden bursts of concentrated nutrition. Trench geography amplified this effect. The V-shaped structure of the Mariana Trench acts almost like a funnel, concentrating sinking organic debris along the narrow trench floor. Instead of being deprived entirely, Hadal organisms may experience long quiet periods interrupted by brief but intense feeding opportunities. The aggressive amphipod swarms suddenly made more sense. By the late 2000s, sustained camera deployments were producing thousands of hours of footage across the Pacific trenches. Similar ecosystems began appearing in the Kermadec Trench, Japan Trench, Tonga Trench, and Philippine Trench. Each trench contained unique species, but the pattern remained consistent. The Hadal zone was biologically active. Marine biology textbooks that once described the deepest ocean as nearly barren were becoming outdated in real time. Then came the moment that pushed the scientific shock even further. Cameras deployed deeper than 8,000 meters began recording something many researchers thought should not exist there at all: the fish that should not have been there. For decades, marine biologists believed they knew exactly where fish life ended. Somewhere below 8,000 meters, the physics of the deep ocean were supposed to become too extreme for vertebrates to survive. Pressure at those depths exceeds 800 atmospheres, enough to compress steel structures, destabilize proteins, and interfere with the chemistry that keeps cells functioning. Scientists expected the deepest trenches to contain crustaceans, microbes, worms, and other pressure-tolerant invertebrates, but not fish. But then all of our assumptions were overturned. International research teams, led by Alan Jameson, filmed small pale fish gliding through the trench. They did not appear injured or dying. They swam naturally, circling bait and moving calmly along the sediment. Later analysis identified the species as the Mariana snailfish, named after British naval officer Herbert Swire, who served aboard HMS Challenger during the original 19th century trench expedition. Mariana snailfish were recorded at depths of approximately 8,145 meters, deeper than any fish reliably observed before. In later years, related Hadal snailfish were filmed in the Japan Trench at depths exceeding 8,300 meters, setting the current verified depth record for fish. These numbers may not sound dramatically different from Challenger Deep's full depth of nearly 10,900 meters, but biologically they are enormous. Every additional kilometer downward produces crushing increases in pressure. By 8,000 meters, organisms exist under conditions that would destroy most vertebrate tissue almost instantly. The snailfish themselves looked unlike typical fish. They lacked the armored scales, rigid skeletons, and muscular frames associated with surface predators. Their bodies were soft and gelatinous, almost translucent beneath camera lights. Some appeared ghostly pink or pale white because their tissues were thin enough for blood vessels and internal structures to show faintly through the skin. Their bones were lightly mineralized, reducing rigidity that might crack under pressure. Instead of bulky muscle mass, they possessed flexible bodies adapted for neutral buoyancy near the trench floor. To human eyes, they looked fragile. In reality, they were among the most pressure-specialized vertebrates on Earth. One reason Hadal snailfish appear so unusual is that pressure changes the rules of biology itself. At sea level, proteins maintain precise three-dimensional shapes required for metabolism, muscle contraction, and cellular repair. Extreme pressure distorts those structures. Cell membranes become less stable. Enzymes stop functioning efficiently. Nervous systems struggle to transmit signals correctly. For years, researchers believed vertebrate biochemistry simply could not operate beyond a certain depth because the molecular machinery of life would begin failing faster than evolution could compensate for it. The key adaptation that changed this understanding was a compound called trimethylamine N-oxide, better known as TMAO. Deep-sea fish accumulate TMAO in their tissues to stabilize proteins against pressure-induced deformation. In simple terms, it acts like a chemical shield helping cellular machinery maintain proper shape under enormous compression. Scientists studying Hadal snailfish discovered they contained some of the highest TMAO concentrations ever recorded in vertebrates. The deeper the fish species lived, the more TMAO researchers generally found in its tissues. This pattern suggested a direct biochemical relationship between survival depth and protein stabilization. But TMAO introduced another mystery. While the compound protects proteins, it also changes the internal chemistry of cells. Beyond certain concentrations, it begins creating problems of its own by interfering with other biological processes. Researchers, including Mackenzie Geringer, studied Hadal snailfish physiology and concluded there may be a hard biochemical ceiling for fish life. Based on pressure calculations and TMAO accumulation rates, many scientists estimate that vertebrate fish may not survive much deeper than roughly 8,200 to 8,500 meters. Below that point, maintaining enough TMAO could itself become physiologically impossible. That theory explains one of the most important details often misunderstood in documentaries and viral videos. Fish are not living at the absolute deepest point of Challenger Deep. The trench floor extends nearly 2,500 meters deeper than the deepest confirmed fish observations. At the very bottom, researchers primarily observe amphipods, sea cucumbers, worms, microbes, and other invertebrate life forms. Yet the snailfish still survive astonishingly close to what may be the vertebrate limit. In evolutionary terms, they are operating at the edge of what fish biology can physically tolerate. The trench footage also revealed behaviors nobody expected from animals living under such extreme pressure. Mariana snailfish did not appear slow or barely functional. They actively hunted small crustaceans and amphipods around baited cameras. Their movements were smooth and controlled despite temperatures hovering just above freezing. Scientists realized these fish were not accidental survivors pushed into the trench by currents. They were fully adapted predators, occupying a stable ecological niche within Hadal ecosystems. The discovery triggered a wider reassessment of vertebrate evolution in extreme environments. Genetic studies showed that Hadal snailfish evolved from shallower relatives over millions of years, gradually adapting to increasing depth. Natural selection repeatedly produced vertebrate fish capable of surviving pressures once considered incompatible with complex life. These fish live at a depth of 8,000 meters, close to what scientists believe may be the absolute biochemical limit for vertebrates. Beyond roughly 8,200 to 8,500 meters, pressure likely disrupts proteins and cellular chemistry faster than organisms can compensate. The trench continues descending another 2 kilometers below them, into regions apparently dominated only by invertebrates and microbes. Interest in the Mariana Trench intensified even further in 2012, when filmmaker and explorer James Cameron descended alone to Challenger Deep aboard the Deep-Sea Challenger. Spending hours at nearly 10,900 meters, he described the seafloor as eerily lunar and almost alien. Later, explorer Victor Vescovo and the Five Deeps expedition used the DSV limiting factor to capture the clearest footage ever recorded from the trench floor, documenting amphipods, sea cucumbers, and active Hadal ecosystems in unprecedented detail. The missions also revealed something disturbing. Plastic waste and microplastics had already reached the deepest place on Earth. Contaminating ecosystems scientists had barely begun understanding at all. There is wonder, and then there is concern. Well, that's it for now. Thanks for watching. Did you like this video? If you did, please consider liking, dropping a comment, and hitting the subscribe button so that you don't miss out on our new uploads. Also, don't miss this video you see on your screen right now. It's truly unbelievable.